Good morning, dear student. Today we will discuss about the isoenzyme, its definition and basic concept. So let's start. So in this topic, what we will study? We will study the definition of isoenzyme, its characteristics, few examples, its mode of origin and some related terminologies, and the methods for identification of the various isoenzyme or their separation method. So let's study one after another. So what is an isoenzyme? That means definition. Different molecular forms of same enzyme synthesized from various tissues or genes are called isoenzyme if they catalyze the same reaction. So, two or more enzymes synthesized from various tissues or different genes catalyze the same reactions are called isoenzyme. If these various types of physically distinct version of same enzyme synthesized from different genes, that means encoded by different genes, they are called true isoenzymes. So from that point of origin, they are different cell tissue origin, different encoded by different genes, but catalyze the same reaction. So what are the characteristics of isoenzymes and what are the uses? Why there are isoenzymes? Okay. Characteristics, according to characteristics, they differ in their sensitivity to different regulatory factors. They may have different substrate affinity. That means KM may be different. That some of the isoenzyme has got the more KM value, high KM value. That means affinity is low, whereas other forms may have the low KM value. That means affinity is high. Characteristics example of one such is hexokinase and glucokinase. They both catalyze the reaction, that is formation of glucose 6-phosphate from glucose. Okay, in the process of glycolysis, glucose is converted to glucose 6-phosphate with the help of enzyme hexokinase, and in the liver with the enzyme glucokinase. So hexokinase, which is present in various tissue other than liver, it has got low KM value. That means high affinity towards substrate. What is the substrate here? Glucose. Whereas glucokinase has got high KM value. That means low affinity towards glucose. Why? Is it has its own physiological benefit, because you know that main function of liver is maintaining the blood glucose homeostasis. So it maintains the blood glucose homeostasis. So if glucokinase has got the same KM value, that means low KM value as hexokinase, so whenever liver hepatocytes itself gets some glucose, they will use the glucose for its own energy production. But purpose of liver is to maintain the blood glucose homeostasis. So it will supply the glucose to various tissue and to maintain the blood glucose. When there is sufficient amount of glucose present to maintain all these function in various tissue, maintaining these homeostasis, then and then only liver use the glucose for its own energy production. Okay, so that is why when glucose is high, then only glucose is converted to glucose 6-phosphate in the process of glycolysis in the liver. So the KM value of glucokinase is high, whereas the KM value of hexokinase is low. So substrate affinity is low for the glucokinase and high for the hexokinase. Okay. Other than this, they are different in their cofactors sensitivity or use of cofactors. Some dehydrogenase use NADH as cofactor, whereas some others use NADPH. So prefer NADPH over NADH as cofactors. Okay. So from regulatory point of view, from substrate affinity, from cofactor utilization, they may be different. So they have different characteristics. That is why, due to this type of differentiation, they are very useful to understand the diseases of different organ, because they different organ prefer the different isoenzymes. So for understand or diagnose the disease of a particular organ, sometimes this isoenzyme is of great help. Okay, and why there are multiple forms? Nature doesn't like wastage, isn't it? Our physiological system doesn't like wastage, so there must be some reason, and one of this reason, other than this differential distribution due to different function of the particular organ, these isoenzyme provides backup copy for emergency purpose. So some essential enzyme nature prefer to produce some backup copy, which gives some time, gives some scope to cope up with some diseases, or in some emergency situation, urgency, to produce this backup copy of some essential enzymes. Okay, so we have told that two isoenzyme originated from different gene, and they arise through gene duplication. These isoenzyme, they may occur in same species, maybe in same tissue, even maybe in the same cell also. So different copies may present in same species, not all the copies may present in a particular individual and a particular individual, but all the copies present in the population. So some has got type A, some has got type B, some have got type C, or a particular individual may possess all the types in his system, but in the different tissue, or even in the same cell, different types of isoenzyme may present, and they have got similar but not identical amino acid sequence. You know that the protein structure is, primary structure of protein is determined by the sequence of amino acid. If the sequence is different, then there are slight differences. So they have similar, very similar amino acid sequence, but not identical. And why that this clearly points towards the evolutionary origin of this enzyme. That means they have common evolutionary origin. That is why they have similar sequence, but due to the need, natural process or natural selection, they develop they gradually develop slightly different sequence of amino acid to produce this isoenzyme. So if we sum up, they generally differ in the kinetic property, regulatory property, their cofactor use, or their subcellular distribution. Okay, they may be soluble or they may be membrane bound. Kinetic, that means Km may be different, Vmax may be different. Regulatory property, their regulation may be, sensitivity towards the regulator may differ in different isoenzyme. They differentially choose for their cofactor. I have already given example of dehydrogenase, and their subcellular distribution may be different. Some may be soluble, some may be membrane bound. Okay. So among the isoenzyme, one very important to mention is lactate dehydrogenase. This is the first isoenzyme ever found, first enzyme found to have isoenzyme. This LDH has got at least 5 different isoenzymes, and all the isoenzyme present in each individual. Okay, so it is the example of isoenzymes, different isoenzyme present in a same individual but in different tissue preferentially. Okay, So how they separated? They separated by electrophoresis. We will study the separation methods later in detail. Okay. So this LDH isoenzyme, it contains 4 polypeptide chain. 4 polypeptide chains. Okay. In hemoglobin, they have got the 4 polypeptide chain, but they are heterodimer, 2 alpha, 2 beta, isn't it? Here also 4 polypeptide chains, and they are 2 types of polypeptides. One is called H for heart, another is called M for muscle. Just like hemoglobin has got alpha and beta, here it is denoted as H for heart, M for muscle. But they combine differentially. How? Some has got the 4H version. They are homopolymer of H. Homopolymer of H. Like heart. In the heart tissue, there are 4H. Okay. Whereas in muscle, they are homopolymer for M. That means all the polypeptide chains are M variety. Okay. So H to M from heart to muscle, and in between these two there are HM, HMM. So 5 types of isoenzyme present differentially in the different tissue for the lactic dehydrogenase. Let's see. LDH1 is called 4H and present in heart and erythrocyte. LDH2, here also the portion of H polypeptide chain is much more than the M. So it is triple HM, also present in heart and erythrocyte. Now comes to 50-50 HHMM. LDH3 is HHMM, present in brain and kidney. Brain and kidney. Please remember, heart and skeletal muscle is easy to remember. Here, in between this, there is brain and kidney. Then gradually portion of AM is increasing and H is decreasing. So LDH4 is HMMM, present in skeletal muscle and liver, and all the polypeptide chains are M varieties present in skeletal muscle and liver. Okay. So these are the different composition of LDH1 to LDH5, and according to this, these are the tissue distribution. In general, the distribution of different isoenzymes of a given enzyme reflects at least 4 factors. What are they? Number 1, they point towards the different metabolic pattern of different organs. Suppose taking example of glycogen phosphorylase. Glycogen phosphorylase is an enzyme which is used for breakdown of glycogenolysis. They are different in skeletal muscle and liver in their regulatory property. The regulation is different. Even in the sensitivity towards the particular regulator is also different. So the glycogen phosphorylase enzyme is different in the point of regulation in the skeletal muscle and the liver. We will discuss it in the during the regulation of glycogen metabolism. Here just know the example, the glycogen phosphorylase. Okay, and you will relate this in your glycogen metabolism class. Okay, then the isoenzyme may different in the same cell. The location of the isoenzyme may be different, and it also reflects the different molecular roles of the different organelles in the same cell. Taking example of isocytic dehydrogenase, they present in the cytoplasm and they may present in the mitochondria of the same cell. Cytoplasmic form of isocytic dehydrogenase is different from the cofactor sensitivity, the uses in the metabolic function than the mitochondrial isocytic dehydrogenase. Number 3, this is very important. According to the stages of development, from the embryonic to the fetal tissue gradually towards the adult tissue, pattern of isoenzymes may different. Some isoenzyme may present in the embryonic and fetal tissue, completely absent in the adult one. They completely changed in the adult form. The isoenzyme is completely other one. Okay, so this is very important for diagnosis of malignancy, carcinoma, because some isoenzyme, suppose embryonic antigen or some human chorionic gonadotrophin, alpha fetoprotein, they present in the fetal form, embryonic form. Okay, but in the malignant cell, they reappear. Some of the malignancies, they reappear, or their level increased in high amount, and by diagnosing this pattern of re-expression of the fetal form or embryonic form in the adult tissue, we can diagnose the malignancy, or even in the severity of malignancy by denoting the quantitative estimation of this type of enzyme or protein. Okay. In case of LDH, the fetal form which present in the liver is completely different from the adult form. Okay, so they denotes towards the developmental stages of our life. And last, they differ in their response to the allosteric regulation. Suppose glucokinase and hexokinase. Hexokinase is more sensitive towards the feedback inhibition of its product glucose 6-phosphate. Glucose 6-phosphate is inhibit the enzyme hexokinase, whereas glucokinase is very less sensitive or insensitive to inhibition by glucose 6-phosphate. So the allosteric regulation sensitivity towards the different regulator is different, may be different in different isoenzyme. Okay, so they, what are the factors? They may points towards the different origins of the gradual development of the stages. They are different in the same cell but different organelles. They may be different in different organs, different towards the substrate sensitivity, towards the regulator sensitivity, and towards the allosteric modulator sensitivity. Okay. Now, isoenzymes may be formed by several ways. You already know the true isoenzyme, that means they are synthesized from the different gene, and classic example of this is salivary and pancreatic amylase. They synthesize from the different genes located on the different chromosome. Okay. Now, what is alloenzyme? Here, they are synthesized from the same gene, even from the same locus, but synthesized from the different alleles. And you know that particular allele is present in particular individual. That is why only one form will be present in one individual. In case of alloenzyme, only one form will be present in one individual, but as a whole, total population share the all forms. Okay, all the forms present in the total population, but for single person only one form is present. Just like the glucose 6-phosphate dehydrogenase, there are various forms, almost 400 different forms of glucose 6-phosphate dehydrogenase has been identified, and they present in the whole population, but particular individuals share the only one form. Okay, just in contrast to lactic dehydrogenase, we have already discussed that 5 isoenzymes all present in same individual. Okay, now what is isoform? What is isoform? Isoform is not isoenzyme. Remember that isoform is not isoenzyme. What is isoform? You know the central dogma, isn't it? In your 10+2, that means DNA to RNA to protein. DNA, synthesis of DNA means replication, then RNA, mRNA means transcription, then protein means translation. Okay. These isoenzymes produced after protein is synthesized, that means post-translational. How? By post-translational modification. So, molecular heterogeneity of an enzyme which produced after protein is synthesized by post-translational modification are called isoform. Example is alkaline phosphatase isoenzymes, bone isoenzyme, liver isoenzyme, kidney isoenzyme. They differ in their sialic acid content. You know the sialic acid. What is sialic acid? N-acetyl neuraminic acid. Okay, NANA. So, by post-translational modification, the different sialic acid content is different amount of sialic acid is added to alkaline phosphatase to create these isoenzymes. So, they are actually isoform. Okay. Different types of isoform may be present in the same individual, just like lactic dehydrogenase isoenzymes, creatine kinase isoenzyme. Just like that, isoforms may be present in same individual. Now, identification of isoenzyme. How do you identify or separate the isoenzyme? First and foremost is electrophoresis, because some of the different isoenzyme have different mobility in the electrical field. So by utilize these facts, the different mobility in the electric field, we can separate them in the electrophoresis, either by polyacrylamide gel electrophoresis or PAGE, or by agar gel electrophoresis. So lactic dehydrogenase, creatine kinase, alkaline phosphatase, they may be separated by electrophoresis. Heat stability. You know that all the enzymes are protein in nature, and protein may denaturate due to heat, isn't it? But some of the proteins may have the more sensitivity towards heat, so they destroy easily or denature easily towards in the heat. Example, bone alkaline phosphatase, and some may be relatively heat stable, so they need more temperature to degrade it or to denaturate. One example is placental isoenzyme. So bone isoenzyme and placental isoenzyme, they may be separated by heat. Okay. First, if you gradually heat the enzymes, first the bone isoenzyme will be destroyed, and after increasing the heat, then placental isoenzyme may destroy it. Okay. By sensitivity towards the inhibitor, some of the isoenzyme may sensitive to one inhibitor and some may not. Just like ACP, acid phosphatase, some acid phosphatase are tartarate labile, some are resistant. So, tartarate labile acid phosphatase may be separated from the stable acid phosphatase with the ACP with the help of inhibitor. Okay. So, electrophoresis, heat stability, inhibitor. Now, they also different in their kinetic property. Suppose Km value, that means substrate affinity or substrate specificity, may be different for different isoenzyme. We have already given the example of glucokinase and hexokinase. They may also differ for their cofactor requirement, like dehydrogenase. Some dehydrogenases are NAD plus dependent, some are NADP plus dependent. Tissue localization may be different. We have in detail discussed the tissue distribution of the LDH, and same is true for creatine kinase, alkaline phosphatase also. Different forms, CK1, CK2 and CK3 are differently located in the different tissue, and also the alkaline phosphatase also. And sensitivity towards the specific antibodies, they also may differ. Some are sensitive to a particular antibody, whereas some are not, and this is the this property can be utilized to separate the isoenzyme. And classic example is creatine kinase, which can be separated by using specific antibodies. So, for lab identification, we can use the electrophoresis, heat stability or heat denaturation process by use of inhibitors, by use of Km value, identifying the Km value, and by use of specific antibodies. And if you do the core research or the analytical purpose, then it can be also different, separate or identified by the use of cofactor and their tissue localization. Okay, so these 7 can be used for the separation or identification of particular isoenzyme. Okay, so these are all for isoenzymes. So you have to remember the definition, some example, their properties, how they differ. In short note, you have to write this and how they can be separated. Okay, that's all for today. Thank you. Hope you all are doing well. So study well. We'll see you soon. Bye.